This is for WJEC, GCSE Biology, section 3.2 as part of the Human Health and Disease section. Lymphocytes and phagocytes. I want to talk to you about how these two cells are used in the immune system to defend ourselves against disease-causing microorganisms. Now, a disease-causing microorganism, because you should be aware that not all microorganisms cause disease, but those that do are called a pathogen. Now, pathogens will have on their surface structures called an antigen, which I'm representing here with little triangles. I'm trying to use triangles because you need to understand that the shape of the antigen is crucial to how the immune system works. Antigens are found on your own cells as well, so immune system will come across one of your own body cells and it would not recognize it as being foreign because you have what we call self antigens. So these are foreign antigens on these foreign microorganisms, and if they're causing disease, they're called pathogens. Now, in this particular example, I'm demonstrating with a bacteria, but don't forget other pathogens will include fungi and viruses. Now, the problem with bacteria is they make you feel ill because they release toxins. And as they release these toxins, they can affect your cells, possibly cause cell death, and that's why bacterial infections can be quite harmful to you. Now, what we need to do is that we need to use an immune cell that is going to respond to both the pathogen, in this case a bacteria, and the toxin, because we need to stop the toxin harming you. Now, the immune cells that are involved in this are what we call lymphocytes. What you're seeing coming up on the screen here is a selection of different lymphocytes. Scientists estimate that you could have maybe up to 10 million different types of these lymphocytes. I'm only going to show you 5 here. But how they differ is in those little structures you can see projecting from the lymphocyte here, which are called antibodies. Now, antibodies are specific in their shape, and their shape will mirror the shape of the antigen or whatever they need to bind to, what they need to stick to. So you can see here I've got 5 different shapes of antibodies. They are specific in their particular shape. We need lymphocytes here that are going to respond to the pathogen with its triangular shape antigens here, and the toxins that they are producing, those little oval shapes here. So there are 2 lymphocytes in here that I would say are much more suited to responding to this particular microorganism and the toxins it's producing. The 2 flashing now are the ones that I'm trying to show you that they would respond to the pathogen and the toxin simply because they have got a shape that mirrors the antigen and the toxin. So the moment they are stimulated, in other words, these lymphocytes have been found and they are triggered to respond to the infection, what they'll do is that they will divide and copy themselves rapidly. I'm not going to show that here, but they would make masses and masses of the same lymphocyte here, all with the same type of specific antibody. But how they now respond is that each one will start secreting these antibodies. We are talking rapid secretion here. Scientists estimate that they're probably releasing about 2,000 antibodies per second, and they're just going to flood your bloodstream with antibodies, and they're going to move around your body as the blood circulates around your body. And what they do is that they will stick to anything that they have a shape that they mirror. Let me show you that on the next slide. So here we've got our pathogen with its triangular antigens, and here we've got our toxins. So what happens is that the antibodies that recognize the toxins bind to the actual toxins, effectively neutralizing them. Those toxins now will not be able to bind to your cells, will not be able to enter your cells, will not cause any harm to your cells. These antibodies have neutralized the toxin. This is where they get a change of name. They are still antibodies, but because they've bound to the toxins, we call them antitoxins. The problem still exists, however. We still have these bacteria that are producing the toxins, so we need to stop those now as well. While the other lymphocyte was producing antibodies specific to the antigens on this pathogen. So these antibodies will stick to the antigens. They do a number of things here. They may well destroy this pathogen directly. They will certainly immobilize it. In other words, this bacteria is not going to be able to move around. And if it was a pathogen that enters your cells, such as a virus, it would actually stop that virus from entering your cell as well. But they do have one other role as well, and this is a role that includes the other type of white blood cell in the immune system. The other type of white blood cell in the immune system are the phagocytes. So here's our pathogen surrounded by the antibodies that have stuck to the antigens. And phagocytes are triggered by the presence of antibodies. So as they move around the body, if they come across an antibody, what they start to do is they start to move their membrane and the cytoplasm around the antibody and the pathogen they're attached to. The technical word here is they engulf. And by engulfing, they take the pathogen and the antibodies that are stuck to the antigens on the pathogen inside the phagocyte. Here what they do is they now digest the actual pathogen by releasing digestive enzymes. And they will break down that pathogen, destroying it. So that will deal with infection. But what you might have noticed in your life is that once you have a particular disease, you can develop some immunity to it. Or if you're being given a vaccination, which sometimes actually contains a disease in a very low dose or a harmless dose, you have immunity to it. So you may never even realize if you've caught the disease. Let me explain why. In initial response, you found your body used lymphocytes to produce antibodies against that pathogen. Those lymphocytes that were used remain in a high number inside your body, perhaps for the rest of your life, certainly for a number of years. Because they remain in your body for a long time and they are specific to that particular disease, that particular pathogen that affected you, we call them memory cells. It's almost as if your immune system has a memory of the disease that you originally caught. So they're a much higher number than before, which means if the pathogen infects you again, the same pathogen you had before, it is much more likely for these lymphocytes to be stimulated to do what they did before. And what they were stimulated to do before was to release antibodies. But because they're in greater number, they will produce antibodies much faster and in much greater quantity than before. Which means your pathogen is going to have these antibodies stick to it very, very quickly before it can probably reproduce quickly and before it can maybe even produce toxins. So you may never even realize that you've had an infection once you've had one before, because you now have immunity to this particular disease.